Good morning. I'm going to present to you the results of a very interesting project that was carried out in our premises and aimed at the evaluation of the genetic variability and reproduction in meagre broodstocks. This work has been carried out at the Hellenic Centre for Marine Research, which is the main research body working in marine sciences, and it employs more than 500 scientists in at least 3 distinct locations in Greece, in Anavissos in Attica region, the island of Rhodes, and at Gournes near the city of Heraklion in Crete. More particularly, the Institute of Marine Biology, Biotechnology and Aquaculture has approximately 130 scientists working in Crete and in Attica, and has past and ongoing projects and collaborations in more than 70 countries around the world. Now, the project aimed at increasing basic genetic knowledge for meagre Argyrostomus regius, a species which is considered as emerging and promising for the aquaculture industry across the Mediterranean region. On the top right graph, you can see that this species belongs to the Cyanidae family, with species that are not only fishing targets, with a capture production of around 50,000 tons, but also for the aquaculture industry with more than 200,000 tons. More specifically, we see at the lower right graph that meagre has nowadays reached an aquaculture production of more than 14,000 tons, and this is believed to grow in the near future. Then, our second aim was to use this genetic information to explain the different sizes in meagre, and finally to be in position in the near future to identify quantitative trait loci related with commercially important traits in the species like growth, disease resistance, and others. Towards these objectives, our primary goal was to develop an efficient, reproducible, and cost-effective genetic tool in order to first characterize genetic diversity in captive meagre broodstocks, and then to infer for the first time parentage assignment for fish of different size kept in different sea cages. Therefore, one multiplex PCR set designed with 10 microsatellite loci previously developed for a phylogenetically closed species was optimized and used to genotype and genetically characterize more than 250 meagre samples coming from 8 different stocks of the commercial company. We calculated basic parameters like the number of alleles, the observed and expected heterozygosity, linkage disequilibrium, and also deviations from the Hardy-Weinberg equilibrium across all samples by measuring FIS values and the differentiation among the stocks with the FST value quantified. On the top right image, we show the factorial correspondence analysis for meagre, in which we propose the clustering of the different individuals in 4 distinct groups. This is also evident in the lower right graph, in which we follow the Bayesian approach with the structure software to infer the most likely population structure based on the microsatellite data, and we also saw these 4 different groups of fish that have different colors in the graph. The second objective was then to study parentage in some 400 meagre fish that were sampled in May 2016 and compare these results with another data set of fish that were sampled in January of the same year. We know that due to size differences, fish originating from the same spawning event of a broodstock of 6 females and 13 males were size graded and kept in different sea cages, one for bigger fish that was fished out in January, and one for slightly smaller fish that were fished out in May, and this is the one that we studied. In our study, there was the first successful parentage assignment in the species in meagre with over 90% on average, at an industrial scale production system. Results show that fish belong to 20 families and almost half of the broodstock was identified as probable parents. More particularly, it was 5 out of 6 females and 7 out of the 13 males that participated in the offspring. Because some broodstock fish were injected with GnRH implants, we see that we do not find any significant differences between breeders that were injected and those that were not. From the broodstock that participated, certain parents contributed more offspring and exhibited dominance in spawning. And when we compared these two sea cages, the January and the May one, a slight differential composition for the same families were encountered, as we show in the graph. The 15 shared families that the offspring were assigned to were analyzed for statistical significant differences concerning body weight and total body length differences. Heritability estimates for body weight and total body length were different in the offspring of the different families that were analyzed, and the January batch showed higher heritability estimates than the May batch, and the genetic correlation estimates were almost the same for both species. Regarding now the target market of our research, besides the aquaculture geneticists for whom this research will provide useful information for future QTL identification, the target market is mainly the aquaculture industry and meagre farmers, because in the future, the identification of genomic regions associated with differential growth in the species will provide the necessary tools for marker-assisted and genomic selection. Farmers can then produce same-age meagre with less variability in size. The economic impact of the present study is coming from the application of genetic markers to go for bigger fish with less variability in size and growth, which will lead to a more profitable meagre farming sector. Second, we know that with the future assistance to the industry, we may provide and identify genomic regions associated with differential growth in the species and provide the necessary tools for marker-assisted and genomic selection. Finally, the development of more than one successful breeding programs in meagre is envisaged to produce not only bigger, but also more resistant fish. Concerning meagre, we know that this is a relatively new species for the aquaculture industry, and therefore it is found in the technology readiness level, TRL2, which means technology concept formulated, and it is slightly above TRL1 of basic principles observed. No intellectual property rights have been applied to our output, and we are not planning to do it. Finally, our output is ready to go to the market and be used by the industry. You can find more information of certain aspects of the research that I showed to you in this scientific paper published earlier this year by Nusias et al. In the Aquaculture Journal. Thank you for attending this video. If you like to have more information of the work being carried out in the species, please do not hesitate to contact me at the email address that I provide you. And please, you're more than welcome to visit our brand new website. Thank you again.